Today is topic 14, and this topic I want to introduce a new CAR therapy. It's about a new candidate. They modify a macrophage become like CAR T, and validate the functional and how can it do some anti-tumor function. Okay, so Nani? Okay, it's fine. Okay, so let me briefly introduce what is immunotherapy. This immunotherapy is a kind of cancer treatment. Its concept is use your own body's immune system to find out and further to destroy the cancer cell. So currently, we have several methods for immunotherapy, like monoclonal antibody or cancer vaccine, and some like immunomodulator as some drug for the checkpoint inhibitor. And one of the most popular therapy is a kind of cellular therapy, like CAR T. So what is CAR T? CAR T. Let me try again. Okay. Me? Okay. Okay, so what is CAR T? CAR T full name is called chimeric antigen receptor T cell. So the abbreviation is CAR T cell. And now currently they develop around to third generation. But all these different generation CAR T, their concept is the extracellular domain is used for recognize the specific antigen on tumor cell surface, and the intracellular domain is used for activate T cell. So when the CAR T cell, the extracellular domain binding to the tumor cell, it can activate the T cell to do like cytotoxicity to kill the tumor cell. Okay, so in clinical, what did they do for the CAR T cell therapy? First, they will collect the peripheral blood from the patient and isolate the T cell, and then they will use lentivirus to infection this T cell and insert the CAR gene to the T cell, that this kind of T cell can express the receptor to recognize the tumor specific antigen, and then they will expand this kind of CAR T cell in lab, and after that they will validate the function of this CAR T cell, and when they check this T cell is good for check this T cell quality is good enough, and they will introduce this T cell back to the patient. And in patient's body, this kind of CAR T cell, they will goes following the bloodstream and goes to find out the tumor cell, and the binding and activate, then kill the tumor cell. So since 2017, now they are 6 kind of CAR T cell therapy has been approved by FDA, but most of them are treated for like blood cancer, like the lymphoma or leukemia, and some of them are for the myeloma. And okay. Okay, so this diagram is briefly to describe whole the flow chart for the CAR T cell production. And as I previously described, they will use lentivirus to insert the gene into the T cell and let this T cell overexpress the receptor. And this receptor currently they develop to the 3rd generation. So when the receptor binding to the tumor cell like this, binding to the tumor cell, and this kind of T cell will activate. And not only just cytotoxicity in CD8 T cell, but also they will secrete some cytokine as interferon gamma. It also can influence, that also can impair tumor cell, let the tumor cell goes to cell death. So what did I do for validation of the CAR T cell? There are 3 different methods. One is first they will confirm the the receptor, the chimeric receptor expression on the T cell surface. So this kind of confirmation, they will use flow cytometry to confirm that, because if the chimeric receptor can express on the T cell, it can stand that, surface stand that, and use the flow cytometry to detect it. And the second is for the interferon gamma secretion, because when if the CAR, this receptor, okay, if this receptor express on T cell, and when this kind of CAR T interact with the tumor cell, it can activate better than the original T cell, because of this intracellular domain. Okay, so this kind of CAR T cell can secrete a lot of interferon gamma. And the third function for the CAR T cell is very important, is like cytotoxicity. And they will co-culture the CAR T cell with the modified tumor cell and confirm whether this CAR T cell can kill this tumor cell, like leukemia or myeloma. And after the validation, they will let the CAR T cell go back to the patient's body. So it sounds like a very great method for kill tumor cell, but it still has some limitation of CAR T, because as I mentioned, you can see in currently the FDA approved CAR T for therapy is for lymphoma, leukemia, and the melanoma. It's very, the efficiency is very low in solid tumor. So because CAR T cell is, when the CAR T cell go back to the patient, it's very hard to penetrate into the solid tumor. So it's become the limitation of CAR T cell. Okay, this is the first reason for why they want to find a new candidate for CAR. Okay, and the second problem is the immunosuppressive, because in the tumor microenvironment, there's some kinds of immune cell as regulatory T cell or MDSC. They can suppress CAR T cell activation. So even even the CAR T cell can go to the solid tumor when they when they contact to regulatory T cell or they mate the NDSC, their activity will obviously decrease. So the group want to find out a new candidate for generate a new CAR. Okay, so this is why they choose macrophage, because there are some features in macrophage, like it can localize and persist within the tumor microenvironment. And it's very important. And the second reason is macrophage is important to regulation of the immune system. It's not only just like kill the bacteria, but macrophage also can modulate the immune system for other immune cells. And the third reason is macrophage is a great professional antigen presenting cell. It can present the antigen to CD4 and the CD8 T cell. And the fourth reason is it can participate in immune response activity through phagocytosis and the clearance of cellular debris. Okay, so that's why they choose macrophage to the new candidate against solid tumor. Okay, so let us briefly introduce macrophage. Macrophage derives from monocyte, and in generally, they will go to these two pathways. One is for sometimes it depends on the different condition, because when the condition give the monocyte some cytokine or different antagonist stimulation, it will becomes like M1 macrophage, and this kind of macrophage will express MHC class 2 molecular or like CD80, CD86, or CD68, or iNOS. And this kind of macrophage can secrete some pro-inflammatory cytokine as IL-6 or TNF-alpha, and this kind of cytokine can promote inflammation and activate the whole immune system to kill the target like pathogen in generally. Okay, but sometimes if the monocyte in the like IL-4 or IL-13 conditions, it will become like M2 macrophage. And this kind of macrophage will express CD206 or arginase 1. And this kind of M2 macrophage secrete a cytokine are totally different to M1 macrophage. They can secrete like IL-10 or TGF-beta. But this kind of macrophage, their function are not kill the pathogen or any target cell. Their function is like immunosuppression or clearance the damaged tissue. So maybe you guys can imagine what kind of in tumor microenvironment, what kind of macrophage we found. Many research found in the tumor microenvironment, because based on the tumor microenvironment cytokine or some cell type, it will stimulate the monocyte become like the M2 type macrophage. And in tumor microenvironment, we call this macrophage tumor-associated macrophage, and their features are very similar to M2. They also express CD206, arginase 1, and like this immunosuppressive cytokine. So their functions are very similar, and this kind of macrophage can suppress immune response. So this phenomena is not very good for our immune system to kill the tumor. So this group, the author, they want to generate a kind of M1 macrophage, and they want to modify the M1 macrophage become the CAR macrophage, and put them back to the patient, kill the first thing they want to, this kind of CAR macrophage can kill the specific tumor cell, and the second one, they hope this kind of macrophage can influence other macrophage become M1 form macrophage, even they has already become to like M2 macrophage. So this is the paper today I want to introduce for you. And okay, so they developed some human chimeric antigen receptor macrophage. It's CAR macrophage for cancer immunotherapy. Okay, so what did they do for the CAR macrophage? This kind of structure is based on the CAR T, so you can see it's very similar to CAR T structure, and they only replace like here because they want first they want to try some antigen can express on the tumor surface like CD19 or HER2, and the other one they modify the intracellular domain become Fc gamma receptor, because this kind of domain can activate macrophage, and they packing this plasmid into the lentivirus and then infect into THP1. Because first step, they want to establish the system. So at first, they just try the human macrophage cell line. Okay. And so this is the diagram to show when the CAR receptor expressed in the THP1. It's like that. And if this SCFV domain can express anti-HER2, it means it can recognize the tumor cell surface express HER2. And if this one showed anti-CD19, it means it can against the like leukemia, some B-cell type lymphoma. Okay, so in this research, how did they validate the CAR-M function? They did, in this paper, they did several experiments to validate their CAR macrophage, but most of the important functional assays like, okay, first one is this kind of macrophage need to have the strong phagocytosis ability. And the second is they want this kind of macrophage has the M1-like phenotype. Okay, and the third is important is because as I mentioned previous, we said macrophage is a professional antigen presenting cell. So they want this kind of macrophage can do the very great antigen presenting cell and recruit T cell into the tumor microenvironment and further to kill the tumor cell. So first, just briefly introduce the method they used. Okay, how did they assess the phagocytosis ability? So in whole this paper, they do different type phagocytosis assay. Sometimes they will use the flow cytometry to analyze the phagocytosis. And sometimes they will directly see that, like use microscope-based analysis system. And they have some live image. If you guys are interested in the live image, you can go to their website and you can see whole this process, how the CAR macrophage engulf the tumor cell. Okay, so the first, they try the CAR system in the human monocyte cell line, THP1. And they test different, they introduce different CAR in the THP1, that the THP1 can recognize different kinds of marker on the tumor cell, like CD19 or mesothelin or HER2. Okay, and all of these kind of tumor cell can be recognized by the CAR macrophage, and so as their phagocytosis assessment, you can see the CAR macrophage can engulf more tumor cell in here. Okay. Okay, so because first step they found, okay, they can modify the macrophage become CAR macrophage in the cell line. So the most important thing is they want to modify the primary human monocyte. Yeah, because if you can modulate the primary human monocyte, this method can really used in the patient. Okay, so they change the virus vector, because here they said this kind of adenovirus, this one AD5F35, this adenovirus is more safety than the antivirus, and the infection efficiency is good enough. So okay, so now when following experiment, when they want to infect the primary human monocyte, they all use this kind of virus, and the structure of the CAR are very similar to previous. Just they replace the intracellular domain back to the CD3 epsilon, because they found, okay, even because they found even it's called CD3 epsilon, but it's the structure very similar to FCF gamma receptor, and it also can activate macrophage. So in the primary monocyte, they use this kind of intracellular domain. This is different to the cell line. Okay, but other parts are very similar to previous design. Okay, so let's see the result. And in the primary macrophage, they just collect the PBMC from patient. No, I forgot that is the patient or health people, maybe the health people. Okay, I think it's health people's PBMC, and purify the CD14 positive monocyte, and try to differentiate to the M1 macrophage. After that, they use the adenovirus to infection the M1 macrophage. And the final, they say this kind of CAR macrophage is M1 form macrophage. And then they want to confirm whether this macrophage can express the CAR. So you can see they use the flow cytometry to analyze this, and it can, I think I remember it's around nine people's monocyte, different people. Okay, different, and all of these monocyte can express the CAR. Okay, CAR receptor. Okay, and now they want to validate the modulate macrophage. So here they still do the phagocytosis assay and co-culture the primary macrophage we can call that CAR macrophage. Sorry. Okay, so they co-culture the CAR macrophage with different cancer cell, like breast cancer cell or ovarian cancer cell. And then you can see this kind of CAR macrophage are very specific, to recognize the tumor cell which can express HER2, because if the tumor cell cannot express HER2, the macrophage, even compare the CAR macrophage with the control macrophage, the phagocytosis ability are very similar. But if they co-culture the tumor can overexpress the HER2, you can see because the CAR macrophage can specifically recognize this kind of tumor cell, and so the phagocytosis ability is very great. Okay. And they also test the dose, whether this kind of expression, the CAR expression, the CAR expression level is dose dependent and the time dependent. So they co-cultured macrophage with the tumor cell at a different ratio, and then you can see in different ratio, like this means effector. Effector is called the CAR macrophage, and the target is the tumor cell. Okay, so this 10 to 1, it means 10 macrophage to 1 tumor cell, and this means 1 macrophage to 3 tumor cell. Okay, so in this range, the macrophage phagocytosis ability seems very great. Okay, and so it means the CAR expression level is dose dependent. And they put this co-culture experiment to 60 hours, and you can see following the time point increased, the CAR macrophage phagocytosis ability still very well. Okay, so they demonstrate the CAR macrophage expression CAR is dose and time dependent. So, okay, previous is are the in vitro experiment, in vitro functional validate, and now they want to try some in vivo function. So here they use the ovarian cancer cell and co-administrate with the CAR macrophage by IV, and administrate into the immunodeficient mice. And after several days, around 3 months, okay, around 3 months, and go to observe the tumor growth and the mice survival rate. And as the picture showed, you can see, okay, even finally all the mice died, but the CAR macrophage prolonged the survival in the mice. And when they sacrifice this, when they analyze these mice, they found if the mice co-administrate with CAR macrophage, the tumor burden obviously decreased. So it means the CAR macrophage is used for the to eliminate the tumor cell, especially like the solid tumor, because this kind of tumor cell is the ovarian tumor. Okay, and they also want to see the macrophage function in metastasis. So this is the same experiment with this experiment and just want to analyze the metastasis, and so this kind of tumor can metastasis to lung. So they also want to see whether this macrophage can infiltrate to the lung, the metastasis tumor. And okay, we can direct see the result. Okay, they found less tumor cell in lung, and also can see many CAR macrophage infiltrate to the lung tissue. But if they didn't administer the CAR macrophage, the tumor growth, the tumor metastasis is more severe, and very few macrophage in here because they are skin mice. Okay, and if they administer the control macrophage, it's okay. The tumor is still there, and also less macrophage in the lung tissue. So here they demonstrate, okay, the CAR-M can reduce metastasis burden because the CAR macrophage can infiltrate to the lung tissue and to eliminate the metastasis tumor cell. So next, they want to see some other feature for the tumor feature. And I think this concept is very similar to metastasis. It's called carcinomatosis. Sorry, carcinomatosis. Okay, it's very similar to metastasis, but a little bit different is carcinomatosis. It means at the same time, all the tumor cells spread to different organ. But metastasis, it means, okay, from one tissue or one organ to another one, not at the same time spread to several tissue. Okay, so here they use intraperitoneal injection to see this phenomena. Okay. And this is very similar to previous. It's called administer the ovarian cancer and the CAR macrophage. And you can see, okay, so it's also, this CAR macrophage can reduce the tumor growth in whole body. Because that is, they use IP method, and so the tumor cell will goes everywhere. And it also can prolong the mice survival rate. Okay, so it seems like a great tool for reduced tumor burden in metastasis and the carcinomatosis. Okay, so next they want to check, okay, if this kind of macrophage looks so great, can then exist in the body for a long time, and where are they go? Only just go to the target tumor cell, or can this kind of macrophage can go anywhere in the body? So now they only administer the CAR macrophage in the mice. These mice, they don't have any tumor; they are tumor-free. Okay, so and this kind of macrophage they modified, so this macrophage can express luciferase, and they use IVs to detect the where did this macrophage go. Okay, and first you can see this CAR macrophage can live in mouse bodies for around 2 months, like that, around 2 months. And they also can detect the macrophage in mice body, like this. This picture is for 10 days. And this picture is for 21. And here is the liver. This position is liver. So they think the macrophage after injection, this macrophage they goes to liver and then goes to other tissue. Okay, so they transfer, they all xenograph. Okay, they xenograph 5 different kinds of tumor cell in the ski mice, and administrate the CAR macrophage. And you can see if they the color are very similar. Okay. If they transfer the CAR macrophage, this macrophage can go to tumor. But many CAR macrophage can go to tumor in different kind of tumor cell. And most of these macrophage, and some of these macrophage, they goes to liver, like the previous picture showed that. Okay. And some of the macrophage, they will go to like spleen or lung. Okay. So these experiment, these 2 experiments showed, okay, first is the CAR macrophage can live in the body for like 2 months. It's a very long time for mice. And the other is it can go to several different tissue. And they also goes to liver. Okay. So finally, they want to confirm whether these CAR macrophage are M1 form macrophage, because previous even they do some experiment like functional assay want to validate the CAR macrophage, but it's like on just we can see some feature like a surface marker or cytokine secretion, but they want to use some tool to see the comprehensive transcriptome data. So they here they analyze the CAR macrophage which express HER2 and compare with the control macrophage, and the results show the CAR macrophage can highly express several genes, which genes are associated interferon gamma associated gene. And next, because they want to know not only just the CAR macrophage themselves can express some cytokine and do the phagocytosis to kill the tumor cell. They also hope the CAR macrophage can affect the whole tumor microenvironment immune system. So here they do the different model and use the model for single cell sequencing analysis. And how did they do this model? So first, this is also a kind of ski mice. They don't have a mature immune system, and they transfer human hematopoietic stem cell into these mice and let the hematopoietic stem cell develop to several kinds of immune cell in the mice. And then they subcutaneous inject the ovarian tumor in these mice and let the tumor growth for several days. And then administrate the CAR macrophage in these mice. Okay, and then and then they harvest the tumor cell and you take these whole tumor cell to do not cell, the whole tumor mass, to do the single cell RNA sequencing. So first, let us see the CAR macrophage in the tumor microenvironment. Okay, they can cluster different population to the CAR macrophage and the control macrophage. You can see they are very different group. And to analysis the gene expression, you can see like, okay, because they are CAR macrophages, so only the CAR macrophage cluster can express the SCFB. Okay, and because this one is the control, and this is CAR macrophage, and all of them are macrophage, so they can express CD68. And but the important is because the CAR macrophage, they are M1 form macrophage, so only the control macrophage they can express CD206, and the CAR macrophage they are not. Okay. And the most important thing is the CAR macrophage can express all the interferon associated gene like these three. And they also cluster the tumor microenvironment, exclude the transferred CAR macrophage. And you can see the cluster one is for the the cluster 0. Cluster 0 is for the CAR macrophage tumor microenvironment, and the cluster 1 is for the control macrophage environment. And they found if they put the CAR macrophage in there, the tumor microenvironment can express several MHC are different to the control cluster. And not only just the MHC type are different, but also they also found a whole list tumor microenvironment in the cluster 0. They can express some pro-inflammatory cytokine gene. And then, because they found by the single cell RNA sequencing, they found this CAR macrophage can influence the tumor microenvironment immune system. So they do the in vitro to double confirm this phenomena, and then they co-culture the CAR conditional medium with different cell type, like M2A macrophage or immature dendritic cell or T cell, and then analysis their some gene expression or marker or function, and we can directly say the result. And this they use the CAR conditional medium with the M2 macrophage can influence this M2 macrophage become M1, because this kind of macrophage they can express some, it's very small, okay, it can express some TH1 pathway gene, or like inose signaling, or like interferon gamma. And for the dendritic cell, they also can express some marker, and this marker means this DC toward to inflammatory state, and also can activate T cell to chemotaxis. Okay, so they prove the CAR macrophage indeed to influence the tumor microenvironment immune system become M1-like, and it go to pro-inflammatory state. Okay, and really final, okay, really final. They want to approve the same concept. So let me see what's the experiment. Okay, they do this. This is a kind of in vitro data, and they co-culture the CAR macrophage with tumor and the human M2 macrophage, and to see the okay, and to see the cell death. Okay, cytotoxicity ability. No, not cytotoxicity ability. It means phagocytosis. Okay, and we can directly say the result, because when this group and these three group, when they added CAR macrophage, and even they put co-culture with M0 or other M2 macrophage, if you have the CAR macrophage, it can kill the ovarian tumor cell. Okay. So let us summarize the paper. They generate a new CAR macrophage, and this kind of CAR macrophage can do some phagocytosis function to the tumor cell. And the second is, okay, it can induction M1-like phenotype. Not only this kind of macrophage himself can express the M1 feature, but also it also can influence whole tumor microenvironment immune system, become pro-inflammatory state. And the third is, this kind of macrophage can enhance T-cell cytotoxicity ability. So all of these functions can help to modulate the tumor microenvironment and enhance anti-tumor response. So what is the next step? Okay, so this group say they want to look at selective combination therapy like radiation or immune checkpoint therapy. And so they hope the CAR macrophage can have synergistic effect in the combination with these two different therapy. Maybe can improve them. Okay, and now the team, they are at the Carcima Therapeutics. Okay, it's a pharmaceutical. Okay, it's also their HER2 CAR macrophage is in the human clinical trial. And the third, okay, they think this kind of tool. The tool they develop. Okay, this kind of tool can become a very powerful tool for kill solid tumor. Okay, so thank you for your listening. This is our today's reference. Okay. Ooh. Any question? Oh, we don't know. They also don't know because they didn't do that. I'm not sure. I know. Recently, for the innate immune system, especially macrophage, the innate memory are popular. It's popular topic, but they didn't do any experiment about that. So we don't know, because this kind of macrophage are modified by the adenovirus, so maybe the phenotype are a little bit different with normal macrophage. Yeah, so I have no idea for this question. Sorry.